Why don't we just introduce ourselves while I'm trying to bring my slides back up. Marianne, do you want to go first?
Sure. Sorry, I was just getting my headphones because I remembered the gardeners come today. So my name is Marianne Martone. I'm a professor of neuroscience at the University of California, San Diego, and I am also one of the co-PIs of DKNet. We're very happy to see you all here this morning.
And Jeff?
Jeffrey Greta, also co-PI at DKNet with Marianne, also at the University of California, San Diego.
And Neil?
Hi, my name is Neil McKenna. I'm an associate professor of molecular and cellular biology at Baylor College of Medicine. I'm also the PI on the signaling pathways project, which is an omics resource for cell signaling.
Great. And Koei?
Hello, everyone. I'm Koei Lin, and I'm the project manager of DKNet. And then I'm also working at the University of California, San Diego.
Great. And Scott?
Hi, my name is Scott Oxner. I work with Neil McKenna in the Signaling Pathways Project as lead bioinformatician.
Fantastic. So this is our team. I'm Monica Wesley. I'm the founder of the Sugar Science. And together with DKNet, the Signaling Pathways Project, and JDRF, along with our other partners highlighted below, the T1D Knowledge Portal, Apaters, Protocols.io, HERN, the JDRF T1D Fund, the Diabetes Research Connection, and the Mouse Metabolic Phenotypic Centers. We're presenting the D Challenge to you all, and we thank you so very much for registering and joining us. So what is the D Challenge? The D Challenge is a collaborative effort of sponsors and promotional partners, as well as resource portals and workspaces. You can see our partners are highlighted here. I just mentioned them in the previous slide, and together these groups are working to create a new way of thinking about looking at data. So funding for the D Challenge is provided by the Sugar Science and the JDRF, or the Juvenile Diabetes Research Foundation. And we thank the JDRF very much for their participation. So what is the Sugar Science? The Sugar Science is a digital platform, and we are aimed at helping the T1D, type 1 diabetes research community connect globally. We hope to continue to grow. We're 1 year old, and we're continuing to grow our connections, our collaborations in the way that we facilitate scientist communication with each other. So let's just talk about the D Challenge. The D Challenge is really, you can do the reading yourself, but it's a community research challenge designed to increase awareness about the rich ecosystems and data, online data findable through DKNet and SVP. And we're asking the community to sort of use these data and resources to develop a very compelling hypothesis about treatment, sequelae, or the etiology of type 1 diabetes. So and it's open to all, novices, experienced bioinformatics people, students, data science, professionals. So we have really a wide open invitation. And DKNet has a whole suite of tools and resources. And Neil McKenna has created, the founder of SVP, has created a consensome, which we're going to provide to the community here. Along with our other partners, we're hoping that this is going to be a very rich environment to explore and develop a viable hypothesis around type 1 diabetes. And we're going to walk through like how this whole process will go, but the hypotheses will be presented to a panel of experts. And those will follow, individuals will follow a standard non-disclosure policies. Finalists will, get consultations with the venture firm, the JDRF T1D Fund. We also have $40,000 in cash prizes that will be awarded to winners to use in their research. And then winners will also be invited to an early application for the DRC funding awards. And we really encourage open science principles, For example, the formal pre-registration of any hypotheses developed. And we're going to work with winners to publicize your contributions at a very high level, sort of preserving your hypotheses as you try to, explore them and further them, but at the same time, sort of showcasing what you accomplished. So in short, the Signaling Pathways Project has inferred really that gene regulatory networks from archived T1D transcriptomic data sets and SPP used T1D knowledge portal SNPs to validate the analysis, and they're offering it to each team to view through their own research lens. So people are coming from different research, styles of research or types of research, and using these DKNet and the T1D knowledge portal and looking at this consensome, we hope that they can develop a new hypothesis. So we're asking, the whole group to really just think outside the box, not even think outside the box, think like there is no box, to really try to be creative and innovative in the way you approach these data sets, and use all the levers, use all the tools to really expand your knowledge about these types of tools, but also sort of be very rigorous about how you're testing these data and exploring the data. So the judging and prizes. So on December 1, each team will submit their hypotheses and full documentation. On the 7th, the finalists are announced. And on the 10th, you'll give a final presentation to judges. It's going to be about a 10-minute presentation with 5 minutes for Q&A. And so you have about 2 months to get things, going and ready. If we want to talk about the judging criteria, it's really going to be all about 4 major things: the impact of hypothesis on your hypothesis that you develop on T1D research, the effective use of all these diverse online resources, not just sort of reading papers or anything, so limited like that, just really using the resources and showing that you did use them. They're going to be judged about whether or not this is, how feasible is the proof of concept going to be coming from developing out of this hypothesis, and whether or not you have the completeness of documentation of what tools you did use. So there's going to be 4 prizes in both US and international categories, and awards will range in amount from $2,000 to $10,000 US dollars. Whoops. Judges are going to be sourced from diabetes and data scientists, as well as stakeholders in the type 1 diabetes research community, including but not limited to the following: DKNet, Signaling Pathways Project, JDRF, HERN, the Sugar Science, T1D Fund, T1D Knowledge Portal, University of Florida, and UCSD. And so I guess I would say one thing to remember is really show your work, that, as you're participating in this challenge, you really need to keep good records in terms of what it is what tools you use, what resources you use, how you derived this hypothesis. So at this point, I'm going to stop sharing, and I'm going to hand this over to Marianne, and she will continue.
I think we could maybe keep the slides up, or?
Sure.
But here we go. I've got them. Can everybody see that?
Yes.
Okay. So how does this work? I think Monica already gave you a somewhat of an overview. And basically, DKNet, the Sugar Science, we've assembled a set of resources that provide a starting point for registered teams to use to design a working hypothesis. And we do want to emphasize, as Monica said, that the hypothesis must be testable, right? So this should be something that you could write a grant application on to say, right, we're going to test whether our hypothesis works. We've provided something called the consensum, which Monica mentioned, and that will be presented in more detail, I believe, next week. I think some of you may have seen materials around it, and this will be Neil and Scott talking about that a little bit later as well. That's going to give you some starting points to think about pathways to target in creating a novel hypothesis. DKNet is itself a gateway for locating other types of resources. So data sets, tools, services, possible reagents that can inform your hypothesis or also help you provide supporting evidence for the hypothesis. We really want to emphasize using multiple online resources, not just one. We all know TEDI, we see analyses of TEDI, but there's a wide range of resources that are out there, and we want you to explore those and use them. Again, DKNet and the resources we're providing are a starting point. We aim to be comprehensive, but it's a big world out there, and there may be things that you discover on your own. And actually, we would appreciate knowing about those that we can make DKNet more complete. We want you to avoid heavy reliance on the literature. Of course, in order to see if it's a novel hypothesis, you need to be up to date on the literature, but we really want you to use these data tools and services that are online. That will be one of the main judging criteria. The breadth and depth of engagement with these tools will be important beyond Google and PubMed, which are scientists, of course, two favorite tools. As Monica said, you'll need to carefully document your approach. We will be talking about how you will each set up a protocol space in protocols.io so that you can keep very careful notes. But DKNet also has a lot of materials about how you manage data, especially thinking about new mandates that are coming out with from funders about data management and sharing, and also the FAIR data principles. So we really do want you to think about this, not just a one and done, but how you take the data and analyses that you are working on and manage those as if you were trying to comply with some of these new mandates and engaging in good data management and FAIR practices. There are two places that we've set up on the sugar science. There's something called DKNet Academy, and that has various tutorials on how one can actually use some of the features of DKNet. I think that those will also be presented in some of our check-ins. And then DKNet itself has set up just a little section that says discover T1D resources through DKNet, where we've pre-composed some queries and things and lists of things for you to be able to use. And these include resource reports on tools and resources that are available, databases, projects, organizations, and more. And then the Hypothesis Center, where we feature some of the bioinformatics tools. The Discovery Portal, where we provide search inside of multiple databases that are around the web. So all of these things may be useful starting points for you to find out the different resources that are available to you. DKNet also has an extensive webinar series where we feature different tools that are of interest to the DK community, and those are all online too, and may be useful starting points for you in your research. I also want to call out that we host something called the T1D Research Network, which are a lot of clinical resources that are available to you. So please, you can go to it at this link, and you can also get it off the DKNet homepage to be able to start your exploration. And I wanted to emphasize this again. The reason why we want to do this is, again, a lot of people know about TEDI. They know about GEO, but there are a lot of other resources that are available, and some of them are featured here, like Nanonomy, which has high-resolution electron microscopy data of T1 diabetes. There's knowledge portals. The Monarch Initiative has information about models that span multiple species. But we also really want to emphasize that the answer to T1 diabetes may be someplace else, and it may be in expanding out your search, not just to type 1 diabetes specific resources, but looking at other ones so that you can do comparisons and other things. Right? We really want to say that it's not always type 1 diabetes against control, but type 1 diabetes against a host of other issues that plague us. So we want you to really think about being creative in the way you use and think about online resources for your hypothesis. Okay, and then I think, Jeff, it's on to you. Do you want me to show the slides, or you want?
Yeah, I mean, you can keep the slides.
Okay.
So, you know, as you know, Monica and Marianne have discussed, I mean, you know, there's two goals. You know, the goal of developing the hypothesis, but as Marianne just mentioned, you know, we're trying to increase awareness of, you know, the rich ecosystem of online tools and data that are out there. And so, you know, that's why we're really trying to get all the teams to engage with, you know, this rich ecosystem, and to also bring in, as Marianne said, tools, information sites, that you may be aware of, that, for example, we don't have listed. The important points are that these projects then also have to demonstrate, the value of the public data and the bioinformatics tools that they used in terms of the write-up of the hypothesis. So the documentation of the hypothesis is not just going to be the hypothesis itself, but also the workflow, the steps, you used in terms of the bioinformatics tools used to develop that hypothesis. And that really is meant to demonstrate sort of the fairness of your approach, right? We want all this documented in a way that potentially future researchers could tie online resources together. Next slide. Okay, so getting into a little bit of the administration. So for each team, there is going to be a team lead that Monica will be contacting. There's some other team members that may be useful to each team. So, for example, a team administrator. So if there's someone who can help with setting up, if there's Git repositories that need to be set up, or if there are any technical issues or any technical things that need to be set up, you can also assign a team administrator that we can contact. And then also, ensuring that each team has an appropriate writer or set of writers to both work on the hypothesis, but really to document the process in terms of the tools, the resources that were being used as part of this challenge. Next slide. Okay, so in terms of relevant software and technology, if any of the groups do need a GitHub space, we can set that up for them if needed, or if you want to use your own repo, that's fine. For communications, we are going to be setting up Slack channel. So the registration ended last night, so Monica will collate all that, and then we'll set up the Slack channels in this first week. And we will also be setting up Google Drives for each team, spaces for each team for use for the presentation slide decks, any other documentation for the challenge. I mean, again, these Slack channels and Google Drive spaces are going to be team specific. And if any member of a team, needs a different email to be used, for example, for Google Drive versus Slack, please let us know. And when we reach out to the team leaders, if there are any other team members that aren't registered, that need to be added, please let us know as well. We've also set up a general help email, dchallenge@dknet.org. So if you do have any general questions, that will go into our help desk, and we will get back in touch with you. So that's sort of the overview of just the basic tools. Next slide is fine. So again, one way that we want you to sort of document your workflow, your protocol, is with protocols.io. And so protocols.io allows for free accounts to be set up. So you can set up, your own a free account for creation of the protocol, and then these can be later shared. We will be having at one of the following check-ins, we will be going through protocols.io a little bit, and also talk a little bit more about the final sort of documentation presentation package to help clarify that and answer any questions. But the next set of tools is want to talk a little bit about DKNet and some of the tools that are provided there, and give you a feel for the site itself. And so, the main page will allow you to link out to information about the D-Challenge DKNet. But if you go to the next slide. want to basically provide some background. So DKNet is funded by National Institute of Diabetes and Digestive and Kidney Diseases, and was really meant to be an information portal for biomedical researchers, and to connect DK researchers with NIH-funded resources. And so that's, really what we're trying to do, and why we're using this challenge to also help build out sort of what DKNet provides to the community. Next slide. Marianne already mentioned a number of these services and information places we provide. So next slide. I'll just go into a little bit overview, in the sense that, we're trying to provide researchers with information at various stages of the scientific research life cycle. So Marianne had mentioned the discovery portal for searching for information from various data, information, and other resources. And so, we're providing, these tools to the research community, to assist them with their ongoing research. And next slide. The one area that can be of use, as Marianne mentioned, was the resource reports. And so these provide information on available online resources, information about software tools, about various reagents, that might be useful, and these are searchable, and then we provide information about the tool itself, other places where the tool has been used in the literature, but then also this links directly to the research resource identifier, which documents the tool itself. Next slide.
And I think we do mention elsewhere that for all the tools you use, we would like you to provide the RID for it. So we'll be going over that later.
Yeah, I think I have a slide on that. Yeah. Okay, so the discovery portal, as Marianne had mentioned, provides a way to search, around 200 biomedical databases for potential places to look for information. So again, we're not just, we have a set of, initial, tools and data resources, but, we're really looking, for people to expand that and, bring in, as many useful resources as they find necessary. Okay, next slide. And then Marianne had mentioned the Hypothesis Center. So for some of the tools, so for example, for SBP, for HERN, for use of apaters, and some other resources, we do have tutorials that we are providing, and we hope to expand this with more tools and resources as we move forward. And we also, provide, for example, some integrated use cases. So, one of the things that we're looking for in the challenge is to see how different bioinformatics tools can be used together. So, for example, here there's just an example of how, using phenotype information through the mouse metabolic phenotyping centers, how that can be used to drive a follow-on analysis via the Signaling Pathways Project. And so we really want to see how the the set of bioinformatics tools and resources that are available out there, how they can be used together. And so that's what we're really looking for in this challenge. Next slide. Okay, so we know that there's huge growth of available data and online tools and resources that are out there, and that is what we're trying to get a feel for. So next slide. So for example, through one of the partners that we've started the challenge with, HERN provides a number of different resources, from their resource browser to PancDB, and also to the PancreaTlas. So again, within this one resource, there are three very rich resources. Next slide. The T1D Knowledge Portal, which you'll hear more about at the next check-in, provides a large collection of available data sets for you to interrogate. Again, they'll be presenting in more detail, also with more detail on the consensus at the next check-in. Next slide. And then appeters, which provide a way to take Python notebooks and make them useful online tools, also have a number of resources, and providing bioinformatics tools in such a way makes it very easy to take bioinformatics tools developed and make them available to the broader research community. So the Aperture framework is really a great way to do that. Next slide. And then as we sort of talk about other tools available, other resources. So through DKNet, we have a number of tutorial and webinars that are available online that also get into some of the other resources. So for example, for SPP, the type 1 diabetes knowledge portal, these all already have webinars online, as well as some of the other tools such as Apeters and Hearn, that can be made use of. And these are all available through the DKNet page. So next slide. So the timeline, as Monica had mentioned, today is the initial kickoff. The next Friday, there'll be a deeper dive into the consensome and the T1D knowledge portal. And then there are 3 follow-on check-ins. Again, on the October 21st one, we'll definitely be able to answer any questions, about, the use of protocols.io, and clarify anything in terms of, the documentation that is needed. And then, working up to, the December 1st submittal of hypothesis.
And Jeff, just to qualify, because the participants will be on a Slack channel, or they'll have, the ability to sort of reach out through Slack and with specific questions to Neil and others on the team if needed. Sort of in real time, outside of these formalized meetings.
And that's the final slide. And then I think we're going to be turning it over to Neil. Monica, did you want to handle some questions first around sort of the process, or did we want to handle questions at the end?
We can take some questions around process. Does anyone have any questions? If you do, sort of raise your hand, or you can put it into the Q&A. You can put it into the chat, in whichever way. Oh, someone can't see the slides. Can
everybody else see the slides?
Oh, yes.
He can now. Okay, good.
Okay. Yeah, so if there's, does anyone have any questions, just to drop it in the chat? Okay.
Oh, no problem, Leandra.
No worries. Okay, looks like we're set for now, so let's continue.
Okay. Do you want to share, Neil and Scott, or how do you want to do this? There we go. Perfect.
Okay, can you see that?
Looks great.
Right. Okay, Scott.
All right. So I'm going to start off by just doing a real quick kind of summarization of the signaling pathways project, how we build our underlying databases, how we use those for hypothesis generation, and then talking about the type 1 diabetic data sets that we're using for this project. Okay. Next slide. So as we all know, the archived omics data sets rich sources of information on cellular signaling pathways. These are both transcriptomic and ChIP-seq. But the only problem here is that they're not well organized in public repositories, difficult for your naive investigator to use easily and quickly. So next slide. So that's why at Signaling Pathways Project, we have taken transcriptomic and cystromic data sets, and we've tried to put some organization on top of them. And what we've done is organize them into the data sets themselves into node types, and 4 main node types: receptors, enzymes, transcription factors, and conodes, which is kind of a catch-all character for things that don't fit in the first 3 very nicely. The cystromic data, of course, comes from the sequence read archive, and transcriptomic data comes from public repositories, Array Express, and Geo for the most part. Next slide. So we build what we call consensomes from this data. We build both transcriptomatic transcriptomes as well as cystromic consensome. For the purposes of this presentation, I'm just going to be talking about the cystromic consensomes. And this is a way to rate genes by their transcriptional responsiveness to cellular signaling pathway nodes. So what we did was we utilized a project group out of Japan called ChipAtlas. And what they've done is gone to sequence read archive, analyzed all those experiments using a MAX2 protocol to call peaks. And then they've made that all available. What we've done is taken all those peak max scores in-house, and then for every node or IP'd antigen, we have averaged those max scores across all the data sets and then ranked them from lowest average to highest average. And this is where we come up with our consensome percentile. So for example, what we're looking at here is the ATF4 cystromic consensome. It consists of 4,600 some odd transcriptional targets of ATF4 from your least confident up to the ones you have more confidence in here at the top. Next slide. So how do we use those for hypothesis generation? Well, the first thing we've done is we've grouped these into high confidence transcriptional targets, or HCTs. And basically what we've done is for every cystromic consensome, we've taken the 95th percentile, or the genes that fall in the 95th percentile and above, and we're calling that, these are the ones that we have the most confidence that ATF4 is a transcriptional target of ATF4. So ChipAtlas, cystromic consensomes, high confidence targets, there are 961 unique human nodes for which ChipAtlas curated data sets. And so next slide. What we do with these high confidence targets is we do transcriptional target interaction analysis. And so this really consists of 2 things. First, we have our 961 signatures of high confidence targets. And then you take your favorite gene list. And of course, for purposes of this presentation, let's just say we're using a type 1 diabetic samples versus a healthy control. And someone uses their own full change in p-value cutoffs to come up with their favorite gene list. And then what we do is we determine the probability of intersection between these 2 lists. We use this statistical program R and gene overlap analysis package for this. But this process is really illustrated in this Venn diagram here. So on the left circle, let's say we have genes that are upregulated in T1D versus healthy control. And then in the right circle here, we have our high confidence targets for, and I've just randomly picked this, Notch 1, for example. And remember that these are all the targets that were in the 95th percentile or above. So what we do is we calculate, 1, how large is the intersection, if there's one? What's the odds ratio of that intersection? And what's its p-value? In other words, what is the odds that this intersection could have occurred by chance? So just hypothetical, let's say we had a very large intersection, a large odds ratio, and an extremely low p-value. This would tell us that the intersection between these 2 groups was not occurring by chance, meaning it was real. and then it was a large one. And so the hypothesis that we can generate from this is that T1D genes that are upregulated in this experiment are potentially regulated by Notch1 signaling. Now, this is just one. Imagine that we do this again for all 961 cis-stromic consensos we have, and we can use that to build a signaling cascade or a signaling pathway that would describe upregulated genes in type 1 diabetes. Okay, go ahead. Next slide. And so I'm going to switch gears here, and I'm going to talk quickly here about the underlying data sets that SPP has curated for this project. Now, these are all publicly archived T1D data sets, expression array, and RNA-seq transcriptomic data. When we first looked, the vast majority of the data sets were primarily from blood sources. We had a small group that were from pancreas, and then singleton from endothelium and bone marrow. So for this project, we decided to concentrate on those 18 data sets that had something to do with blood. But even there, we noticed that the makeup of those data sets is very heterogeneous. So for example, our largest group of samples were peripheral blood mononuclear cells. I mean, even those, that's 6 to 7 cell types alone. The next largest group were some purified CD14 monocytes. And then we had a number of singletons from monocyte-derived dendritic cells, CD34 positive hematopoietic stem cells, some purified neutrophils, CD3 positive T cells, and one sample was whole blood. But what all these data sets had in common, of course, is they grabbed samples from diabetic patients and from healthy control. All right, you go ahead. Next slide. Could you switch to the metadata? So one of the things that we're going to provide for our data sets is a wealth of description of the metadata behind these data sets, because I'm sure we're all aware that it's very important to know at what stage maybe of diabetes you're dealing with when you want to compare data sets to data sets. So we've, of course, where available, we've archived what the PubMed for the original publication may be, what the accession number is in the public repository for the data set itself. These are our internal data set identifier. Then we have a data set name, an experiment identifier, and then these are all of our T1D versus non-diabetic contrasts that we've created for these 26 contrasts, and this is from the roughly 18 T1D experiments. But we've also put in an experiment description, which tells some surface details about what the cell type was. So next slide, Neil. Also for each experiment, our next metadata tab, we have kind of curated for each data set what we feel the disease state was, what the tissue source was, the samples, and then the subject age, whether they were young or old, the mean age, details such as were the patient's normal glycemic, were they not, were they long-standing diabetes or early diagnosis diabetes. All these data sets were varied across the different data sets. All right, so go back to the presentation. So what we've done to provide is a set of...
Speed up, Scott.
Yeah, okay. So what we've done for each one of these data sets is we used R and Lima to provide a fold change in a p-value at the feature level. For arrays, that's probe sets. For RNA-seq, that would be gene symbol. And we've provided for each one of the contrasts that I mentioned earlier for each one of those data sets. And some of the QC we do is we look at a PCA plot of the expression data, and we also compare our fold changes that we provide with those that may be in the published data to make sure that our direction of change and magnitude of change is all very similar to what investigators provide in their published paper. Next slide. go ahead and skip this, Neil. It's not too important. So initially, we did apply consistent analysis to the transcriptomic data from the T1D data sets. There were a number of reasons why we were running into some issues with this. One is that there were different expression, the individual, for example, the individual probe sets in an array were going in opposite directions. So for one gene, we have probe sets going up and some probe sets going down. Then there's the issue with distinct criteria and composition of the study cohorts. For example, some of the data were from long-standing diabetes, some was from early diagnosis, some were normal glycemic, some were not. And then of course, there's multiple different cell types. All of this combined to make consensus analysis a little muddy. So what we came up with instead, and we're providing, is a novel analytic pipeline that we're calling feature level enrichment analysis, or FLE analysis. And this uses the feature level data for expression arrays, that's probe sets, for RNA-seq, that's the uniquely mapped gene symbols. And what we've done is we've identified two different sets of transcripts that were enriched. The first set were exclusively type 1 diabetes induced. And by this, I mean, these is a subset of genes induced in at least one experiment, but not repressed in any experiment. And the flip of that, a subset that was type 1 diabetes repressed. And those were repressed in at least in one experiment, but not induced in, not induced in any experiments. We then compared the occurrence of genes in these two lists with the data as a whole, and came up with lists that were either enriched in the induced gene set or were enriched in the repressed gene set. We call these our ET1DI for induced and ET1DR for repressed. This is very analogous to looking at similar gene expression across data sets. So I'm going to go ahead and pass this over to Neil, who's going to talk about this method's validation and some use examples.
Hey, thank you, Scott. So our goal, as Scott mentioned, was to use archived type 1 diabetes expression data sets to infer transcriptional regulatory networks that contribute to this disease, and to make these available to you guys to formulate a novel hypotheses around signaling events in this disease. And so these inferred networks are of one of two types, and these can be exemplified using this sort of a conventional representation of cell signaling. So there are the kind of downstream transcriptional targets that are regulated within the cell that carry out various functions, and we can refer to these as T1D factors. And then there are nodes, signaling nodes, such as transcription factors and enzymes that regulate the expression of these targets, and we can refer to these again in kind of broad terms as drivers of type 1 diabetes. And so we wanted to infer from this analysis nodes and targets whose loss or a gain of function might be involved in this disease, but is not yet known in the research literature. So let me focus firstly on the first of these, and so that's the inferred transcriptional targets. So as Scott mentioned, we have the analysis in which we use to identify targets that were overrepresented among features that were exclusively upregulated, or targets that were overrepresented among features that were significantly downregulated. So obviously, the first thing that we wanted, to do here was to actually, validate these. Start with that. So we wanted to, validate these lists against, independent, benchmarks to establish whether they added, value to information that's already out there. And the first approach that we used, as was mentioned earlier, was using the type 1 diabetes knowledge portal that has amassed a large amount of information on alleles that contain SNPs that are associated with the risk of type 1 of diabetes. And so we want to know whether FLE was a reliable resource, and we anticipated that we would expect to see things, targets that exhibit a strong association with a type 1 diabetes. We would expect to see those highly ranked within our ET1 DIs and ET1 DRs. So we retrieved from T1DK a set of 1,000 targets that exhibit a significant association with the incidence of type 1 diabetes. And then we overlaid them on our ET1 DR analysis. And what you're looking at here is the minus log 10p of the enrichment of the y-axis, and the aggregate log fold change over all of the experiments here on the x-axis. And consistent with the reliability of the ET1 DR enriched network, we saw a very strong enrichment of these most highly ranked T1D KP annotated targets within things that were highly ranked within the ET1 DR network. Now, when we performed the same analysis on the ET1 DI, so these are things that are upregulated, and we didn't say the kind of overlap was more marginal, didn't really reach significant. And so we wondered whether this might be reflecting the fact that SNPs that are associated with type 1 diabetes may preferentially represent a loss of function. So in order to come up with another way to validate these lists, we wanted to focus specifically on expression. We identified a study that examined whole blood expression between type 1 diabetes individuals who were treated with the anti-CD3 antibody HOKT3. So here you're looking at, on the x-axis, here is the log fold change between HOKT3 responders versus control, and on the y-axis the minus log at 10p. And again, consistent with the relevant of the ET1 DR enriched network, we saw that that the highest ranked things in this network were actually strongly enriched in things that were upregulated by OKT3, and similarly with respect to the ET1 DI enriched network, things that were highly ranked within this network were overrepresented among targets that were actually downregulated by HOKT3. So I'll move on now to the second network, that's signaling nodes whose gain or loss of function we infer is involved in with type 1 with diabetes. And as Scott mentioned, they're based upon HCT intersection analysis, where we're computing the significance of the intersection between HCTs for a specific node and things that are regulated in type 1. So now, because type 1 is a disease of the immune system, we figured that a reasonable way to validate these would be a functional approach in the context of T cells. And in order to do this, we use this CRISPR screen by Schiffer et al. That identified things that had essential functions within T cells. So what you're looking at here is a scatter plot of all the targets within this screen. You've got the rank here, and you've got the minus log 10p. And what we found was that nodes that we had identified that had strong intersections with both the ET1DI and ET1DRs, these were actually very nicely represented among things that had significant rankings within this screen. And you can see here that there are several nodes here that I've identified will be familiar in the context of T cells, and others aren't. And so this was interesting because we saw the most significant intersection between nodes that had significant rankings with both ET1DIs and ET1DRs. And we think, we figured that a reasonable explanation for this is that a specific node, has the ability both to kind of activate or repress expression. And, both of these functions, can be, within the same node. And we figure that this is going to be relevant both in the context of normal T cells, but also in the context of type 1. Now, my time is kind of limited here. So, but it suffices to say that nodes that have got high rankings within these networks have got known roles within other autoimmune conditions. So I think this kind of augurs well for the ability of our analysis to identify potentially novel nodes within this condition. So I guess we're kind of running near the top of the hour here. I had a few more slides, but I guess we're going to do another webinar next week in which I'll go into the actual files and the resources that we will make available. So with that, I will hand back.
Yeah, that was great. Thank you, Neil. This is a very powerful tool you've developed, and I think that part of everyone on the call is really interested to see that at the next meeting, more of a deep dive into it and how they may use it. So it's really, really exciting, really interesting. You've done a tremendous amount of work, and also hats off to you for using this system in the COVID landscape and actually publishing some very important papers, using this whole construct. So thanks again for that. I see that, several questions have come in, and I think they've all been answered in the chat. But, if someone is interested in asking a question, we can, raise your hand at the bottom of the screen. You can raise your hand and then we can unmute you if you'd like to speak. Or you can just add another, add another question to the ongoing chat. We're happy to take questions, so please don't feel shy. There's a couple of questions about like, can you be a team of one? Yes, you can. We're going to share the slides. We're going to share the video. What else?
Monica, will everything be linked off the D1 challenge, the D challenge page on
Yes.
Sugar Science?
It will be.
And also we want to emphasize, Jeff said we're going to set up Slack as a means of communication. So we will pin a lot of the documents to Slack. So if you want a different email than the ones that you've supplied to us, please let us know so we can set it up with the appropriate email. There will also be a Google Drive that will be set up also for the same thing. If there's a different email than the one you supplied that you like to use for Google Drive, for example, I like to use my Gmail account, just please let us know because we'll have all of that set up by next week.
Yeah, I'll be reaching out to all the teams and just making sure that we have the team lead, the person who's going to be doing the writing and the person who's sort of almost like the admin contact, I guess, or the tech contact. So that'll be coming to you. Keep an eye on your email for that. And how are people feeling? Okay. Oh, wait, here's a question. Can we develop new bioinformatics tools to complement the existing ones in case we need it? What say you?
Yes.
Yes. Yeah, no, I would say, right, the purpose of this is not to develop a new tool, but if you need it to analyze data or, right, take some online resource and augment it, then yes, you could do that. But that's not the deliverable, right? The deliverable is the hypothesis.
Okay, and in the Q&A we've got, do you feel confident that those of us that are basic bench scientists with little to no bioinformatics background will be competent to use these tools? Okay, and I think, what
yes,
do you
absolutely.
Yeah.
Neil, I think, will be available as a mentor, and the DKNet Hypothesis Center is really meant to help guide you into using some of these. And so please reach out. There will be other resources at your disposal where your experience as a bench researcher may really help you, right, interpret those data sets. For example, you see there's a lot of imaging data and other sorts of things. So it's not just about analyzing gene networks. So that's what we really want to encourage you to explore, starting with the consenso. And Neil is a great mentor.
Yeah, I would also just sort of jump on the back of that and say that this whole exercise is really about learning the available tools and diving in and using them and connecting them. And I do think that with Neil and Scott as your guides through this, what they've created in the consensome, it's really going to be a seamless process and a very unique opportunity.
There's another question. Marianne, I think this is you had.
Oh yeah, so Nanonomy, right? So you can explore those types of resources through DKNet. There are actually several imaging databases. This one in particular is called Nanonomy, and they have these huge high-resolution electron micrographs for exploring online, and several of them actually are related to T1D. And so these are the types of resources that one can discover that you might be able to use in very novel ways, yes. And they, as far as I know, are available for analysis. It's freely available.
And this was so yes, we are going to be putting this, another question, we are going to be posting the slides for access by the teams and the video as well.
Yeah.
Okay, and the database of electron images, you got that?
Yeah.
Okay, perfect. Okay, well, it's 10:01.
Yeah.
So we look forward to seeing, representatives from each team at the next meeting. And at that point, we're actually going to have a presentation from the T1D Knowledge Portal team, as well as from the Signaling Pathways Project. So you'll have a really great introduction into those tools, and we'll look forward to seeing you again.
Mm-hmm.
Thank you all for joining
us. All right, you're off.
Thank you.
Bye-bye.
Bye.